can answer them in the Q&A. So next up, we have Daniel DeSouza, and he obtained his Associates of Biology at Northeastern Junior College, and he'll be transferring to CSU in biochemistry.
All right. Hello, everybody. My name is Daniel DeSouza. In this summer, I studied edge effects on soil pH and canopy cover with Claire Winfrey, Noah Fierer, and Julian Rosasco. Before we get started, I'd like to thank everybody in the Fierer lab. They're all awesome guys, girls, everybody. Just showed me so much stuff. I got so much great experience in the lab, and I'm so thankful for that. We can go to the next slide now. All right, so imagine you're in this field and it's a hot day and you need to cool off. You decide to go into the forest to get some shade, but you notice as you're walking, it's getting cooler, even though you're not quite in the forest yet. What you're feeling is basically an edge effect. An edge effect is a diverse alteration associated with artificial boundaries of habitat fragments. An edge effect can be observed in this figure detailing canopy cover versus distance from the edge. As we go along the x-axis, once we hit the edge, we see a drastic spike in canopy cover on the y-axis. Previous work on edge effects in plants has found that edges can affect soil pH, organic carbon quality and quantity, and plant communities and abundance associated with the edge. To date, most of the research on edge effects is focused on how they impact plants and large-scale animals, but edges affect soil as well. Yeah, I totally saw that Asen was supposed to go now, but gotta go. Soil is the foundation for many things we enjoy, anywhere from farming to just enjoying the vegetation that grows out of it. But if you go down to the small scale, there's an entire world, even in a handful of soil. This microbiome is crucial to soil health, and in turn, it's important for the health of anything that depends on the soil. However, that microbiome is in danger. Species loss is a large-scale issue being studied today, and the leading causes of it are habitat destruction and fragmentation. An important way that conservation biologists assess the impact of fragmentation on communities is by observing these edge effects. However, despite knowing edge effects are useful for examining fragmentation and soil health being at risk around the world, there isn't much work on how edges affect soil communities. Next slide, please. So that brings us to the topic of my research. The Savannah River site, or the SRS for short, as seen in the picture on the left, is a giant research site in South Carolina where they maintain meadow patches and forest matrices. We sampled 5 rectangular meadow patches in the SRS in order to see if there's an edge effect on a variety of things like soil pH, microbial richness, etc. In the middle picture, you can see where all of the patches we studied are located in the SRS. To do this, we laid 4 transects spanning a meadow and forest habitat, going along the top, bottom, left, and right of each of the 5 sites. Seen in, you guessed it, the picture on the right. So at these sites, we took samples to get things like soil pH, and we took landscape data to get things like canopy cover. And talking about landscape data, we'll go into, you can go to the next slide. We'll go into that. This figure illustrates vegetation cover represented by the green lines and canopy cover represented by the purple lines. You can see as we go along the transect, canopy cover shoots up as we hit the edge and vegetation cover goes down. To obtain the vegetation cover, we took pictures at a standardized height at each sampling point. And then we divided the pictures up by overlaying a 5x5 grid on top of them and approximated vegetation cover on a scale based off of the amount of green in each segment, with 0 being absolutely no green in the segment and 4 being the segment's just all green. In a similar method to obtain canopy cover, we used a spherical densitometer, which is like a mirror that you hold out in front of you and it reflects the canopy above. And we counted up those segments to get the canopy cover. We can go to the next slide. So the main thing I looked at with the data we gathered was how edges affect soil pH. And this is because pH is the best predictor of soil bacterial richness. The figure on the right details our expected results. The x-axis shows distance from the edge, with the edge being represented by the vertical dotted line. And the y-axis illustrates pH going from low at the bottom to high on the top. We expected to see an edge effect in pH with the rate of change dependent on the distance from the edge, dipping lower into the forest than the meadow due to the acidity of pine needles. And this is illustrated by the solid purple line. Alternatively, we could have seen that pH just got lower in the forest without a gradual change, which is illustrated by the dotted green lines. or the pH would remain constant going along the transect from the forest to the meadow, which is illustrated by the dotted black line. These two outcomes illustrated by the dotted black and green lines would not illustrate an edge effect and would probably be due to other influences. You can go to the next one. So our data ended up showing 3 general trends, but for the most part, we did not see an edge effect. We saw that pH either varied randomly, had very little change, or slightly dipped along the transect. Similarly to the figure detailing our expected results, the 3 x-axes illustrate distance from the edge. This time illustrated by the edge is illustrated by the edge axis tick, and the y-axes illustrate pH on a range from 3 to 8. And the 3 different figures each represent a different meadow patch. The site most in line with our hypothesis was site 2, with the soil exhibiting a slightly more acidic pH in the forest and in the meadow. However, this could be due to other factors and doesn't necessarily illustrate an edge effect. The other sites either looked like sites 1 and 3, some varied wildly like site 1, and some were fairly constant throughout like site 3. Some, the point along the transect didn't seem to matter. It's just, we really don't know why. You can go to the next slide. We don't really know why we saw this, but what we think happened is before these sites were acquired by the SRS, they had a varied land use history. And land use history is a signal of land use history and soil pH can be measured for many years. So these are aerial photos taken prior to the SRS being established, and they detail that previously the land was used for agriculture. You can see in this picture on the left, that is a site with particularly bad land use history. Half of it is a field and half of it is remnant forest.
7 minutes.
You can see on the right that it's fairly good, and that's the site we did see a slight dip. You can go to the next patch or the next slide. So where do we go from here? Next thing we'll do is connect land use history with pH to see how much of an effect it plays. And then we'll investigate our giant data set relating to microbial data and connect that to pH, the pH trends we observed. And open for questions now.
We have time for one quick question. Danny, what effect would latitude have on forest edge effects?
So if the sites were like sloped at all, if there was any like elevation increase, that could affect stuff like pH with water running down the hill. But mostly the edge effects are just between like what we see at the edge between the meadow and the forest, all the differences we see at the edge there.
Okay, great answer. Great job, Danny. Okay, if you have, as I said before, if any audience members have more questions for Danny, please post them in our Q&A and he can answer them during the next presentation.